In 1880, Charles Darwin published his penultimate book, The Power of Movement in Plants, in which he described the way plants respond to external stimuli with adaptive movements, growing towards the light in the case of a shoot, positive phototropism, and in the case of the roots, growing away from the light, negative phototropism, or towards the center of the earth, geotropism. Movement in response to touch, thigmotropism, can be seen with Venus flytraps and with the sensitive touch-me-not plants. This was largely a descriptive work. Darwin realized that movements like the ones he described were perfectly consistent with evolution by natural selection in being adaptive, but he made no attempt to determine the underlying mechanism by which the movements were occurring. Nonetheless, Darwin is credited, usually, with the discovery of auxin, the very first plant hormone ever described, although the mechanism of auxin wasn't elucidated until many years after Darwin's death by Fritz Wendt, and it wasn't until 1937 that its chemical structure was identified as indole 3-acetic acid, or IAA. Somehow, Darwin nonetheless gets credit, once again for being the first, and also perhaps for not having made any huge goofs in his interpretation. In this case, he was the first to describe the phenomenon of a specific manifestation of phototropism, in which a growing seedling of canary grass can be made to curve by shining a light on it unidirectionally from the side. Normally illuminated from above as a seedling popping up from the soil and perhaps surrounded by other plants, the coleoptile grows straight upward. Darwin, actually two Darwins, Charles and his son Francis, showed that by illuminating the seedling from the side, the coleoptile can be made to bend toward the light source. Cutting right to the chase, the mechanism at work here is hormonal. Auxin is produced at the growing tip of the plant, the shoot, apical meristem in the case of U. dicot, and the amount of auxin produced by the meristem determines how much growth occurs. Moreover, the plant adjusts auxin production to induce an appropriate amount of growth. A growing tip that is poorly illuminated needs to stretch upwards or redirect its growth toward whatever direction the light is coming from. In contrast, a well-lit growing tip has no need to stretch and really should not be engaging in light-seeking growth in the way that a poorly illuminated tip would. The less light a tip receives, the more auxin it produces. More auxin results in a greater amount of elongation in the cells differentiating in the stem just below the apical meristem, the primary xylem and phloem, etc. Under poorly lit conditions where auxin is produced at greater levels, there's more cell elongation, while under well-lit conditions where less auxin is produced, the cells elongate less. As a result of this basic dynamic, a plant grown under weakly lit conditions is actually expected to grow taller than one under well-lit conditions because of the greater cell elongation. The internode distances, the length of the stem between the nodes where the leaves attach, are predictably longer, resulting in a plant shape often referred to as spindly or leggy. Well-lit plants, in contrast, have shorter internode distances and are more compact in shape. The tendency of a seedling to bend toward its source of illumination turned out to be a consequence of auxin transported from the poorly lit side of the apical meristem to the cells below it on the poorly lit side of the seedling. By producing more auxin, the cells on the dark side of the growing tip would elongate more compared with the cells on the well-lit side, and thus the plant's tip would bend toward the light until it got into a position in which its meristem was evenly illuminated. And at that time, there would be uniform auxin on all sides of the growth tip, and thus it would grow straight with uniform cell elongation. It should also be easy to visualize why this mechanism is adaptive, why it has been perfected and maintained by most plants over eons of natural selection. In a thought experiment, compare a plant that is able to respond to low light by growing more and a plant in which this ability is disabled. Now, under well-lit conditions, both plants grow to identical sizes and shapes and have equal success in survival and reproduction. What is the fitness advantage of the plant that does have the normal cell response, elongation in response to auxin? It's entirely fair to treat this auxin-based physiological mechanism as being the way in which positive phototropism, movement toward a light source, is achieved in plant growth. And if you're a plant, the advantage of being able to grow toward the light should be pretty obvious, considering that your fundamental metabolism is based on photosynthesis. A weird twist with this particular response, cell elongation near the growth tip in response to auxin, is a way we have turned this against plants with the herbicide 2,4-D, 2,4-dichlorophenoxyacetic acid, an auxin analog, that is, a molecule that looks enough like auxin to fool the plant's auxin receptors, in this case triggering such an exaggerated cell elongation that the cells collapse and the growth tip dies, exposing the plant to pathogens that quickly kill the whole plant. 2,4-D is the favorite herbicide for use in lawns to kill non-grass or broad-leafed weeds, also referred to as forbs, which are usually eudicot and have the kind of growth mechanism that I've been talking about so far. Grasses, being monocots, have a different growth pattern, and their meristematic tissue is enveloped within many sheaths of developing leaves, and it's actually under the surface of the soil, making it well protected from the 2,4-D herbicide when it's sprayed over the lawn. Consequently, 2,4-D acts lethally against forbs, but not grasses, and this is a useful agent for people who, for some reason, want a grassy turf devoid of any broadleaf plants. We started this discussion with a mechanistic interaction between the hormone auxin and its target cells, leading to the response. We then talked about why this response is adaptive to the point of being maintained across many plant taxa, and then we finished with some rationale for how the physiological interaction is exploited for some practical intervention by humans. We're going to follow this template of physiology, adaptive value, and practical application for the remainder of this lecture. Look for these three elements in the discussions that ensue. The second discussion we're having is again about auxin, but this time on its effects on plant branching in the shoot system and also in the root system. Our target cells are the ones that grow into the lateral branches and respond to auxin trickling down from the shoot apical meristem via the phloem. But the action is completely opposite in the shoot versus the root system. Remember from an earlier lecture when we discussed these tiny embryonic shoots that remain in a dormant state at the leaf scars, the axillary buds? Well, these are mini apical meristems that remain inactive as long as there's enough auxin to keep them suppressed. Most of these axillary buds never develop into branches, but the ones that do often respond to a decline in the auxin signal coming from the top of the shoot apical meristem. The most obvious cause for a steep drop in auxin would be the removal of the apical meristem, either by herbivory or by human intervention. The practice of topping a plant by removing the growing tip of the shoot has the effect of causing a more bushy growth pattern, with many lateral shoots rather than just the main one. We do this either to generate a desired shape of the plant, one that's more bushy and compact, or to increase the number of vegetative branches, which could increase the yield of a flowering or fruiting plant. Once lateral shoots initiate their growth, they start producing an abundance of auxin at their apical meristems, thus halting the auxin-poor condition that would allow the induction of development in remaining axillary buds. Basically, if a growing shoot is left to grow unmolested, its growth tip effectively inhibits the development of side shoots, resulting in all of the water and nutrients from the root system being monopolized by the single growing tip. This is the phenomenon of apical dominance, Presumably, this is advantageous for a young plant that may be small relative to nearby established plants, so strong growth upward is a better use of resources than allocation to many shoots in a highly branching bush. When the shoot tip is removed, as with topping, there is no apex to dominate, and so lateral branching is allowed until the auxin production produced by the new shoots reestablishes apical dominance. Again, note that we've covered the three elements I mentioned before: physiology, that of apical dominance; adaptive significance, the allocation of growth resources to the main stalk in a young plant; and application, how manipulation of apical dominance is used in practical horticulture. On the other side of the soil line, auxin influences the rate of branching in the root system as well. Only here the effect is positive. Unlike the inhibitory effect that auxin had on shoot branching, auxin increases the rate of side root formation from the various types of lateral meristem and pericycle tissues. We take advantage of this response by using auxin analogs to induce root development on cuttings, pieces taken from a plant's shoot that are put into water or soil to develop their own root systems and thus make clones of the mother plant. This is a widely used practice for rapidly producing a large number of clones from a desirable plant. The dicey part of this is getting the cuttings to root and to develop healthy root systems quickly. Rooting hormone increases both the success rate and the speed in which cuttings develop into healthy individual plants. The rooting hormone is, another auxin analog. The real auxin is indole-3 acetic acid, and your typical rooting hormone that you can get at Home Depot or Lowe's is indole-3 butyric acid, basically a synthetic substitute for the native auxin, which, if you could get it, would work just as well, if not better. Now we come to the adaptive significance part of the story. And to set the context here, you have to think about the issue of maintaining an appropriate balance of root biomass to shoot biomass. There's an optimal ratio of root to shoot. Too much shoot and not enough roots would mean there's not enough surface area for water and mineral uptake to meet the needs of the photosynthetic canopy. Too much roots to serve a small shoot system would be an unnecessary expense of resources. The plant would have been better served to direct those resources to more growth in the shoot, which would result in more photosynthesis, more generation of additional resources, and presumably greater fitness. This second auxin mechanism of negatively affecting shoot growth and positively affecting the development of additional root surface area is actually a homeostatic mechanism for shoot-root biomass ratio. If something happens to cause an excess of root biomass, for example, if an herbivore or a gardener removes large amounts of the photosynthetic canopy, the loss of the shoot apical meristems, the auxin sources, tips the balance towards shoot regrowth through the development of lateral branches in the axillary buds and against further root surface area increases because there's less auxin to trigger side branching in the root system. On the other hand, any excess of shoot relative to roots would be short-lived because the abundance of auxin produced by many apical meristems would tip the balance in the other direction, favoring the growth of the root system while increasing inhibition of the shoot growth. Presumably, plants that are able to maintain an optimal root to shoot biomass ratio have a fitness advantage over those that don't have this ability, and this basic mechanism has consequently been maintained and refined by natural selection. There are other important hormonal mechanisms working in concert with auxin in this regard. Your text will address the effects of cytokinins, but the general idea is illustrated well enough by auxin. The third hormonal discussion in this lecture is also relating to the hormone auxin, but in this case, the same chemical, indole-3 acetic acid, is produced by the ovarium, the part of the pistil in which the fertilized ovules grow into seeds after pollination. In berries, the class of fruit that contains multiple seeds developing within a single ovarium. A successful pollination results in some number of sporophytic embryos. It could be one or it could be several hundred, as in the case of a kiwi fruit. The amount of auxin produced by the ovarium is proportional to the number of healthy embryos it contains, fertilized ovules on their way to develop into viable seeds and offspring of the mama plant. With great pollination success, there will be many babies and the auxin signal will be strong. If there are just a few babies inside, the auxin signal will be weak. The plant makes use of this information in determining which ovaries it keeps, the ones that will get the sugars and water and other nutrients needed to develop into mature berries, and which ovaries it abscises, drop to the ground unceremoniously after fruit set but before maturation. Because plants always overshoot with the number of flowers and the number of fruits it sets, only to mature fruit from a small subset of them. So that's the physiology, and now how is this adaptive? Well, it should be pretty obvious. If you're a tomato plant and have to choose between putting your resources into ovarium 1 with 30 seeds or ovarium 2 with 10 seeds, you get potentially 3 times the fitness by maturing ovarium 1 and dropping ovarium 2. A plant that ignores this information provided by the ovarium is going to suffer a fitness deficit. Now, the same mechanism can be used to artificially induce parthenocarpy, the maturation of fruit containing no fertilized ovules. And this is done by applying auxin, or auxin analogs, during the window in which the plant makes the decision to mature or abscise a given fruit. Once a plant has made the commitment to mature a fruit, then nothing stops it. A whole watermelon, which is just a very large berry, will be matured even if it contains no viable seeds. This is actually one method that has been used to produce seedless watermelons, but the more common technique used currently is to cross-fertilize a diploid and a tetraploid plant to make triploid seeds, which will undergo parthenocarpy without any chemical intervention. The use of auxin as a fruit set hormone is still common, and the home gardener can get auxin sprays for their tomato plants and kiwi vines at places like Home Depot or Lowe's. I mentioned earlier that the hormone auxin acted in conjunction with a separate hormone, cytokinin, in the maintenance of root shoot biomass homeostasis. Similarly here, auxin as a fruit set mediator operates in conjunction with another important plant hormone, gibberellin. Gibberellins and cytokinins are just about as diverse as auxin when it comes to their actions in plant physiology. And if we had more time, we would pursue them in greater detail. But alas, you're spared the additional 30 minutes of discussion needed to do a minimal degree of justice there. Let the multifaceted picture of auxin as a plant hormone suffice as representative of the ways in which typical plant hormones can work. The one additional discussion that I have opted to include in this lecture relates to the most well-known fruit ripening hormone, ethylene, which organic chemistry students would know by its IUPAC name, ethene, C2H4 with a double bond between the carbons. It's a small molecule hydrocarbon and therefore a gas at room temperature. So the first mold broken by our consideration of ethylene is the idea that hormones have to be large, complex organic molecules that travel through the plant in the phloem. The second mold-breaking realization here is that ethylene operates in a way that is anti-homeostatic, making the fruit ripening process snowball uncontrollably through positive feedback. This happens because ethylene, the hormone that causes fruits to ripen, is produced by ripening fruit. In other words, a ripening fruit releases ethylene, which causes more fruit to ripen. And this results in a much stronger ethylene release, which then causes many more fruit to ripen, which then start releasing ethylene. And pretty soon you have a ridiculous amount of ethylene and massive amounts of ripe and rotting fruit. This is a real challenge for cargo ships with their holds full of tropical fruit like mangoes or bananas. A single ripe banana can trigger a cascade that causes the whole shipment to be ruined in the time it takes to get from the port where it's loaded to the destination port where the fruit are unloaded for distribution to U.S. markets. The trick to prevent this from being a serious risk is to pump large amounts of air through the cargo holds, ventilating out the ethylene gas before it has a chance to affect nearby fruit. The ethylene mechanism is also useful to us when we need to speed up the ripening of an avocado. This can be accomplished by enclosing it in a paper bag. Most supermarket tomatoes are shipped as hard, green, unripe fruit and are gassed into ripeness with ethylene just before putting them out on display. Unfortunately, the flavors of tomatoes shipped and ripened in this way is nothing close to a vine-ripened tomato, but that's just my opinion. Maybe some of you like tomatoes with hardly any tomato flavor. The bigger, more challenging question here is, why would such a non-homeostatic mechanism be maintained by natural selection? Usually it's adaptive to have built-in mechanisms for stability, like with the auxin mechanism for root-shoot biomass homeostasis. This positive feedback system for fruit ripening via ethylene is very common, though not universal for fruiting plants. There are a lot of plants with fruits that don't ripen off the vine in response to ethylene. Citrus plants are a good example. But take my old apricot tree. It flowers early in the spring and sets an abundance of fruit that grow to size relatively quickly but stay fuzzy and green, hard and distastefully sour for a long time. And then, usually sometime in June, a few will start ripening. And then within a week, I'll have way more apricots than I can use fresh. All the rats and the birds in the neighborhood come over to eat the apricots, and then within another 2 weeks, there's not a single apricot left on the tree. Personally, I would prefer to have a few apricots ripen every day all summer long, but this never happens thanks to ethylene. My question now is, how is this a good thing for the apricot tree? We'll talk about this in our class meeting.